Hello, today we're going to talk about meiosis. Interphase 1. DNA replication takes place in S phase. Chromosomes are replicated. Pairs of sister chromatids are made. Chromatids are each of the two thread-like strands into which a chromosome divides longitudinally during cell division. Each chromatid contains a double helix of DNA.
Prophase 1. In this phase is when a cell has two N chromosomes, double chromatid. N is a haploid number of chromosomes. Homologous chromosomes pair, also called the synapses, creating a bivalent. Crossing over occurs. Crossing over is the exchange of genes between non-sister chromatids, resulting in a mixture of parental characteristics in offspring, otherwise known as a recombination of alleles. Crossing over increases genetic variation through recombination of linked alleles. In crossing over, there's three steps. The first one is synapsis. It's when homologous chromosomes associate, creating a bivalent. Then it's chiasma formation, and the third one is recombination.
Chiasma formation is when neighboring non-sister chromatids are cut at the same point. A holiday junction forms as the DNA of the cut sections attach to open end of the opposite non-sister chromatid. Recombination is a result where alleles are swapped between non-sister chromatids. Metaphase 1. Spindle microtubules move homologous pairs to the equator of the cell. Orientation of paternal and maternal chromosomes on either side of equator is random and independent of other homologous pairs.
Anaphase 1. Homologous pairs are separated. The sister chromatids remain attached. This is known as reduction division. A non-disjunction can occur, creating mutations.
Telophase 1. Chromosomes uncoil, and a cleavage furrow is formed between cells.
Cytokinesis. A cell membrane forms around new haploid cells. Meiosis 2. Interphase 2. In this phase, no replication occurs. The nucleus breaks down.
Prophase 2. Chromosomes condense and they become visible. No crossing over occurs in this phase.
Metaphase 2. Here, the spindle microtubules move chromosome pairs to the equator of the cell by attaching to the centromere of the chromosomes. Random orientation happens, which will create variation in the gametes, but not as much as in metaphase 1. The genetic variation that happens in this phase is 2 to the power of 23.
Anaphase 2. Centromeres contract and they separate the chromatids to opposite poles. The non-disjunction can occur, and this would lead to two gametes containing the wrong chromosome number.
Telophase 2. In this phase, chromatids reach opposite poles. New haploid nuclei are formed.
At the end of meiosis 2, cytokinesis happens. It's when the cell membrane forms around the four new haploid gamete cells, and this ends as a result of a diploid zygote.